one of the reasons is I want you not to rely too much on lecture notes. That's because you like to do that. You get all these lecture notes, nobody bothered to open the book. So this is like a start for you because these are actually direct from the book. Okay? I have been going through this book back and forth. It seems like the language is a bit, how to say, it prerequisite style kind of knowledge, meaning that if you compare this book, this physiology book, with the regular biology book that you use during your matrix or pre-university, you know, like Raven, Campbell, Madden, so on, you might find later the sentences used here, they kind of assume that you have known something. Yeah, and I know most students, I mean like our student, they don't really bother to go anything further than the paper. So let's put it this way. Maybe we can reduce the number of chapters that we need to learn. Like, you see here, right? We've got this lesson plan here. So altogether, we have how many chapters? 9, 10, 11, 12, 13. That's 4, 5. Was it 4 or 5? 1, 2, 3, 4, 5. 5. So I'm thinking, let's focus to finish off respiration and photosynthesis at a more relaxed pace. Okay, because unless if your IQ is 3000, I don't mind. We can go super fast. But since most of you, this would be the first time learning such a thing, let's take a time to sit in it and absorb it. Yeah, this can be your reference material. Even if you don't open the book, I hope that's not the case. You can still get the knowledge somehow. Right. So last week we didn't finish it so much. There's one more subtopic that I hope that you have read. Have you read? Sila tak? The final bit of it, the key experiments in understanding photosynthesis. So there's a little experiment. You can see in figure 9.9 how scientists came about the knowledge of, plants use rainbow color to energize photosynthesis. Yeah. So in this case, maybe I can open it quickly here. We get 10 minutes to finish that off. Oops. Stop here, right? Yeah. So using an algae, this Engelmann, T.W. Engelmann, he used this Spirogyra. It's an aquatic alga, filamentous, meaning that they can appear like a strands of something. So that is Spirogyra. So he had this Spirogyra floating about in a pond, and then in the pond he has a light coming towards it. Right in front of the light, he put a prism, and as we all know, prism separate the light into its constituent, especially the visible light. So incident light coming into prism, then the light is scattered into the individual constituent. How many colors are there in rainbow? 7. So all of these 7 colors that actually landed on the water, you can see that the bacteria that is close to the Spirogyra here, remember, Spirogyra doing photosynthesis and therefore releasing oxygen. Bacteria use the oxygen released by the Spirogyra. So in essence, the more oxygen there is, the high population of the bacteria we're going to expect. So you can see that Spirogyra that is close to the blue region and the red region will have the most population of the oxygen-seeking bacteria. This is important, okay, because there are many bacteria that are anoxygenic, meaning that they don't release CO2 and they don't even use CO2. For example, those bacteria in the volcano got bacteria, yeah, thermophile bacteria. Yeah, they use sulfur in the metabolism. Okay. Yeah. Yeah. And so this energy of light actually absorbed by whom? So that's what you need to understand now. Light that coming to the leaf is actually absorbed, and the thing that is absorbed is what that we're going to learn in this coming chapter. Okay, so in general, the thing that absorb light, we call it as pigment. Okay, not all pigments will appear some kind of color to our eyes. Some pigments, they are pretty much colorless. Some pigments, they look black. Well, not really black. Sometimes the concentration of one color is just too much. Okay, for example, you can see that in Cameron Highland or in any horticultural produce, they produce this black tomato, black plum. Actually, it's not black. It's just the high concentration of anthocyanin. It's just too much of it to the point no light actually go through or get reflected back. Yeah. If you don't believe me, go to Tesco, not Tesco now, what you call it, the supermarket, Lotus. On the shelf, you know Ribena? You look at the Ribena bottle, what color do you see? You see black, right? You black. But when you dilute the Ribena, now it gives the beautiful purple berry color. Yeah. The reason it look black, because in the cordial bottle, super concentrated. That's why it appear black. So the same concept to the tomato that you see black, right? So it's not really black, it's just high concentration of one pigment coming together, right? But when you dilute it, the real color will be visible to your eyes. So these light energy will be absorbed by the pigment. There are many pigments in a given leaf. Yeah, even your skin. Our skin here. Don't only think, oh, I only got brown color. No, there are other colors actually. Okay, and these can easily get manifested if you eat a bit more something. For example, carotenoids. Carrot. If you eat too much carrot, you will start to have all this orange, yellowish on your palm, the back of your ear. Not because you're getting jaundice. No, it's just the build up of the pigments. More than your liver can store up, more than your body can metabolize. So it has to go somewhere. Okay. Other organisms are like that as well, like salmon. Salmon. What? Give me one example of fish that the flesh is white and red. I don't eat fish. I don't know. Yeah. Forgot that fish. Is anchovy fish? Right. Okay. So this will be absorbed. Then if the pigments absorbed and there is no energy transition happen to the pigment, still no photosynthesis will happen. Okay. The fact that photosynthesis can happen, meaning that the pigments involved in the process absorb the light energy and then do something with it energy, pass it around. Okay, energy transfer. Okay, your table here, the reason it appears black because or dark brown because it absorbs light energy, right, from this light. But does it do photosynthesis? Why not? The absorbed photonic energy, it's not passed around to any biological molecules. Okay, when it's not passed around to any biological molecules, no biological or biochemical process can take place. And that's why the table cannot do photosynthesis, but the leaf can do photosynthesis, right? Give the notes. Right. Okay. And this is also the concept that you need to understand here, which is the quantum yield here. So we have a little graph here. The number of photons given to photosynthetic cells in relation to the oxygen produced per amount of photon or light particle give. You can see here, as the number of photon increase, meaning that you increase the intensity of the light, more oxygen is produced. But at some point, it starts to plateau off. The curve start to relax and it start to become more and more flat. So the first part of this, by the way, this form of curve, we call it biphasic, two phases. The first phase is the steepest part of it. So we call this the slope, the initial slope of it. And then the second part of it is the saturating phase of the slope. So the first part, the initial slope, is actually how quantum yield is obtained simply by dividing between oxygen over the amount of quanta absorbed. Quanta, do you know quanta? It's plural. The singular is quantum. Quantum means packets of energy. The lights that come to the earth, or even lights in this room, it's not coming to you in one form of energy. Like this light give you, say, 500 nanometer of energy. No, you are actually being showered by small particles of light coming to you. Maybe there's coming to you like 3 trillion of light particles now. Each light particles have one associated energy with it. Okay, 3 trillion, maybe 10% of it blue light, 10% of it red light, and so on. So these individual packet of light of similar energy content, we call it quantum. Okay, right. So the maximum yield, as it is stated here, for one oxygen to be evolved or released, it is needed about 2,500 of chlorophyll molecules. Okay, so that's giving you another perspective as well. When you look at the plant, don't think there's only one or two pigments in there. There are only God knows how many billion, trillions of pigments. Why so many pigments? To capture the light to the best of ability. If there's only one pigment, two pigment, what happens if the light's coming abundantly, in excessive? Who's going to capture it? So losses of energy will occur. So plants try to prevent that from happening. Okay, so this is something that you can read about light drive the reduction of NAP, because we're going to learn this anyway. This is just like to give you the overview of it, how the concept of reduction. I hope you still remember from your chemistry lesson, redox reaction. What's that? Reduction, oxidation. Remember, in science, when something is said to be reduced, reducing, or reduction, it involves the action of giving hydrogen. Okay? For example, I'll give you one example here. You have a substance here. This substance here, without anything, we call it oxidized. Okay, oxidized meaning that it lacks oxygen. lacks electron. But the moment hydrogen is given to it, hydrogen atom, remember it has one electron, one proton. Now it becomes a new substance which is called reduced substance. So reduced simply means one hydrogen has been attached to the molecule. Okay, so that's why you can use this small formula to benda aku merepek. Betul lah kan? So R for reduction is gaining. Gaining who? Electron. O is oxidation is losing. Losing who? Electron. Okay? So when you read articles about, dietitian persuading you to eat more veggie, that kind of stuff, to increase antioxidant intake in you, this antioxidant is actually the reduced form of a nutrient. That nutrient has extra electron that it can donate. Donate to who? Donate to any substance in your body that is unstable. Why they are unstable? Because the electron is unpaired. Remember the law of electron? It must achieve either duplet or octet conformation. It must be in 2 or it must be in 8. Okay? When it's extra electron there, it will start to steal electron from the surrounding molecule. So a cascade of falling apart molecule will start to happen because you steal my electron, I'll steal other people's electron. Stealing upon stealing upon stealing, the whole membrane will be collapsed. Okay, to prevent that from happening, that's why we eat an antioxidant. Instead of stealing electron from the molecules in our body, antioxidant give the extra electron to these unstable molecules so that our membranes, our compounds in the body are undisturbed. Okay, plants got that as well, right? Okay, right. I think we can go straight to the new topic for today. That's for the rest for you to read. Okay. Like I said, I'm going to reduce the amount of lesson that you are undergo so that you can take your time to absorb. Maybe you want to pray at the back. Who knows? Dah baca doa? Okay, so light reaction, okay? We have covered 9.1 until 9.3. Now we're going to continue from 9.4 until 9.9. I do not know, maybe you only finish up to 9.5. Let's see. Number one, what you need to understand is the organization of photosynthetic apparatus. This is very important. This is the basic of photosynthesis. You need to understand photosynthesis is not happening in just about anywhere. It is happening in a specific location inside a cell. Okay? And inside a cell, there are many organelles, meaning that membrane-bound structure. Okay? Subcellular compartment of a cell. Okay? And there are many. There are nucleus, there are vacuoles, there are mitochondria, and so on. We are interested with a type of plastid. Okay, so there is a group of organelles. Collectively, it's called plastid. One type of plastid that we are interested today, which is the chloroplast. Okay, which is this guy. So this is a cross section from electron microscope. You can see that chloroplast. Why is it not green? This is electron microscope, no color. Okay. Actually, the reason you see the color green for the chloroplast is because you are dealing with visible light. When the sample is in the microscope, people are not using visible light anymore. People use argon gas. So no color. But they give off the charges and signals so that the computer can interpret, you get this image. Right. There are important structure that you need to understand here. This is look a bit boring, right? Let's use the cartoon part of it. So in the plant cell, you got this plant cell, see? Many blobs, and then we are interested with these green blobs here. Okay? Enlarge this, and then you get this. Number one, chloroplast double membrane. Please remember that. It's got the outer membrane and inner membrane that enveloped the entire thing. Okay? And in between the membrane, there is an intermembrane space. And this is very important because not all substance can go in and out into the chloroplast. It's kind of highly regulated. Okay? And this is also important because this is where where the plants will eventually decide to have more sugar transported out of the cells or otherwise. Okay? So inside the chloroplast now, you can see the stacking of pancake here, the green pancake here. Yeah. So we focus in here, we enlarge this here. I know that maybe you have learned this before, the concept of grana, granum, that kind of thing. But this time we're going to correct your understanding with the correct labeling. Okay. So a stack of this thing is called granum. 1 pancake, 2 pancake, 3, 4, 5, 6. The whole thing is called granum. Okay. 1 pancake is called a thylakoid disc. It's not written this there, but it's called disc. It's actually a disc. Okay. And this kind of granum, when you go deeper in learning photosynthesis, it's actually called as either grana lamellae or stroma lamellae, meaning that depending on the location and the shape of this structure, it will determine later what kind of biochemical process happens to it. Look at here. This collectively you call it grana lamellae, right? Remember, grana lamellae, stack of thylakoid. This is where the photosystem is located. You will learn later that in order to capture and utilize the light, plants have 2 important photosystems. Photosystem to do what? Capture and utilize. Okay, so the first one of the photosystem is located primarily in this grana lamellae. And what about these connecting bridges here? This is called the stroma lamellae. Okay, it is not part of the pancake, but it's rather protruding out of the structure. Okay. Look at this. This is actually not individual disc, even though I called this just now. It's actually like your intestine. Your intestine is a long tube, right? 6 meters or more. So that tube is arranged in such a way. Does it look like your intestine? So this tube, when it is arranged on top of each other in that folding manner, it gives the illusion of pancake stacking, but it's actually a one long tube like your intestine. Okay, right. So what other thing is important? The stroma. What is the stroma? The stroma is the environment inside the chloroplast, the soup inside the chloroplast. If the soup in the cell we call cytoplasm, the soup in the chloroplast we call stroma. Okay, not stoma. Stoma are the microscopic pores on the leaf surface. That's not it. This is stroma. Okay, right. Yeah. Get it? I do expect for you to be able to draw this. Okay, So I do not know. This is the time, if you don't like drawing, this is the time to like it a bit, right? Okay, moving on. You will find that from time to time in the note, it says to refer to the web essay. This thing is actually available online. I'm going to put it in the folder anyway, so you can have access. So in the web essay, it tells further about the things that you just learned, right? For example, we just learned about the thylakoid, right? Thylakoid, grana, and so on. Look at here. This is the confocal image, meaning that the sample has been dyed with fluorescence dye. You can see here, this is your grana lamellae. What's that bridge? Stroma lamellae. See? And guess what? They are actually all one long tubing. See? It is all connected. That's why I said it looks actually like your intestine. Right? And what about here? Well, here is actually, it says that sometimes in the plastid, chloroplast is type of plastid. There are other plastic that can present as well, like the chromoplast that gives certain petals or fruit the color, right? So, but the important thing is, please understand how the components in chloroplast is organized. Okay, now we go deeper now into the thylakoid itself. So you got your chloroplast, then you got your thylakoid here. Now we go to the thylakoid membrane. So it is a membrane, okay? We are talking deeper, okay? I just want to put things into perspective now. You know the plasma membrane of a cell, right? Now you are inside the cells, inside an organ, inside a membrane. So there are many layers of inside. This is super small. Okay, we are talking about maybe not nanometer, we are talking about picometer right now. So small. Okay, when talk about membrane, thylakoid is also a membrane. The important thing to know is that the thylakoid membrane contains this thing, the integral membrane protein, meaning that the membrane of a thylakoid, I enlarge this, okay? There is a protein in here. This is protein in here. And this is called the integral protein, okay? The membrane is made up of two lipid layer. So this is lipid layer, okay? And the lipid layer, when you enlarge this head and tail structure, phosphatidylcholine, you will have the hydrophobic region, hydrophilic region, water-loving region, and the hydrophobic region. Okay, this thing here. So this thing here is arranged in such a way. There are some part will be hydrophobic, some part will be hydrophilic. This is important because hydrophobic region repels water. Okay, repels water, and it ensures that things do not get solubilized easily. Things do not get merged together like the water, right? You have water, you put one color in, later you get a new color because water is soluble. It makes everything. So this hydrophobic region prevents that from happening, so that other structure can be present in it without being disturbed, right? Okay, so what do I expect you to know from here? Just that the fact that this is integral membrane protein. Some proteins actually only present on the surface. You only get here. Okay, some protein are only on this side. Some protein is actually attached like that. So you can imagine that the surface of a membrane, not only the surface of a cell, any membrane is actually a very busy area. You have imagine that this table is the surface of a membrane. It's full of antenna. It's full of holes. It's full of gates. It's a busy place. It's like Think of something. I'm thinking of a busy, maybe like a, some urban area got boulevard. By the side you got shops, and then suddenly you got your trash can, suddenly you got your hub to wait for your grab to come, and then there is a telephone to call the police in case you got chased by a bad guy. And then there's also a place for you to jump to the next boulevard, that kind of thing. It's a busy place, okay? And that also applied to the thylakoid membrane. Yeah. And then look at here. This is the thylakoid membrane, this thing here, that gets even deeper now. Super zoom. Actually, scientists do not have the image, the actual image for this. This is like a model of it. They tried to get it, but the image still looked fuzzy. I think they have the microscope now to get all the deep in here, but they reserve it for the NASA. They don't want to release the image for us. Okay, so this is a thylakoid disc. You see, it is connected. On the surface of the thylakoid disc, the thylakoid membranes, I want you to know these things. There are 4 proteins that is involved in the photosystems in thylakoid membrane. 4 proteins. Photosystem II, Photosystem I, cytochrome B6F, and also ATP synthase. These are all proteins. Remember, you have learned about the photosystem, right? Also, photosystem II, photosystem. What is photosystem actually? Photosystem is actually protein. It is a protein, okay? It's not alone one protein, no. It is a main protein structure, and then other subunit of other protein attached to it to make it functional, okay? So you can see in this image here, these are the labeling. Photosystem II, 1 protein, cytochrome B6F, 1 protein, photosystem 1 protein, ATP synthase, 1 protein. Then you will see the dimer here, LHC1. LHC means light harvesting complex 1, meaning that this subunit of a protein is meant to be attached to, since the number is 1, is meant to be attached to photosystem 1 here. That's why the labeling become like this, the cartoon version of it. You have PS1 protein flanked or sandwiched by a pair of LHC1 dimer. Dimer means the subunit got 2 components to it. Di means 2. Mer, in science, mer, like polymer, monomer, and so on. The word mer means building block or smallest unit of something. So it can be anything. Monomer, polymer, dimer, trimer, and so on. M-E-R, yeah? Not M-U-R. Murmur. That's not it. Okay. And another thing that you need to understand here about the localization of the protein. This is very important. Localization of the protein. You can read a bit more later. I can highlight here is that, remember that the lamellae means filaments, because this tubing looks like a filament. Lamellae. You got 2 types of lamellae now. Grana lamellae, stroma lamellae. That's what you have here. The middle region here is the grana lamellae. This the one that is stacking here. 1, 2, 3. Grana lamellae. Then you got the edge of it before you found the stroma lamellae. It's very important to understand here that photosystem and the other 2 proteins, the ATP synthase and cytochrome B6F, they have what we call as protein localization, meaning that these 4 proteins do not appear randomly on the lamellae. No, they are actually concentrated in one places. For example, like this one, photosystem II. Photosystem II, you will find them predominantly in grana lamellae. This one here. This, what color is this? Ketayap, kuning hijau. I don't know. Can I complain to this book? Do the proper colouring. Kau nampak apa? Tak nampak. Tak tahu lah, warna apa? How to call this colour? This is green, this is also green. Greyish green. Greyish green, brighter green. Yeah. Bluish green. Okay, bluish green. So the bluish green, that is the photosystem II, you will see that they are only present in the grana lamellae. Predominantly. Okay. But the photosystem I, together with its associated light harvesting complex, LHC1, they are predominantly found in the edge of grana lamellae, but more and more in the stroma lamellae. Very important. Aku tak belajar kan semua ni. Sebab aku belajar pun aku tak ingat. Tak tahu. Yes. However, for the cytochrome B6F, in here, what color is this? Cyan. I call it cyan. For the cytochrome B6F, the cyans, you can see that it's pretty much everywhere. It's here on the grana lamellae, edge of grana lamellae, and also the ATP synthase. ATP synthase is also present in a random distribution. Okay, so why? This is important so that the energy transfer between these protein, by energy I means electron, they are in the most efficient manner ever. Because this thing, all these photosystem, they will be activated somehow, meaning that they receive energy. In order to ensure that they get activated in the most efficient manner possible, they need to be positioned in the strategic location. That's the reason, okay? Imagine if, let's say, I can make a hypothetical situation as well. Okay, imagine if this photosystem I, PS1 protein is located right in the middle of the folding here. Meaning that it's kind of dark and gloomy space in there. It will not have access to the final molecule, which is present in abundance in stroma. You see, photosystem I, at the end of the road, it needs to pass the electron to an electron acceptor molecule. This molecule, by the way, it's called NADP plus, P plus. It needs to pass eventually to this thing. This thing will accept and becomes NADPH. This NADP plus, it's in the stroma. Stroma is this region. The what color is this? Yellowish, light yellowish region. If it's in here, can it has easy access to NADP? Not easy. Not easy. So inefficiency will occur. That's why the design has been in such a way, you photosystem I and your light harvesting complex stay on the edge of grana lamellae, or even better, straight in the stroma lamellae. Get exposed to the soup of the chloroplast so that when you are ready, send electron to an ADP right away. And then you'll get your reducing molecule. Okay. This is another representation of it. Okay. So this is a schematic diagram of it. Remember, this is thylakoid membrane. Membrane, phospholipid bilayer. Okay. Why it's called phospholipid? That thing just now, it's called phosphatidylcholine. Phosphatidylcholine. You got your head here, you got your neck here, you got your tail here. Okay, so the The head here is the phosphate. In the middle here, you got your glycerol. And the tail here, you got your CH, the hydrocarbon tail, right? So that's why the membrane is called phospholipid. Phospho referring to the head, lipid referring to the glycerol. Phospholipid bilayer, two layers of it. Okay, and in between this phospholipid bilayer, there are integral protein. These are the integral protein: PS2 protein, PS1 protein, cytochrome B6F protein, and also the ATP synthase protein. Okay, you can see that they are arranged in a vectorial way, meaning that in sequence, in the right positioning. You are not going to find this PS2 protein in between cytochrome and PS1. You're not going to find it because the energy move from here to here. That's how. If the water flow from the waterfall to the river to the sea following the gravity, this thing is follow the energy transfer from the high energy to the low energy, meaning that when you are talking about the energy content of it, this side of the membrane dealing with higher energy because they just receive it fresh from the sunlight. High energy. As you go further to this side, less amount of energy. Okay. Right. Have you seen this before? This diagram? Yes, you have. Stroma, the soup of it. Now, learn another thing, which is lumen. Not lumos, that's light. Lumen is the inside of the thylakoid membrane. So you got your membrane here, the inside of it. Yeah. It's kind of like your intestine. The inner of your intestine, that is the lumen, where you got all your villi, your microbes, bacteria, and so on. The outside of it is the rest of it. Okay, right. So for proteins, please do not forget that. Down the line, I know we're going to see it in a bit, but I'll just give you the a heads up about the terminologies. You're going to see this thing as well. PQ, PH, PC, FD, FNR. So these things are we call as electron carrier. Okay, there's two concepts in biochemistry when it comes to molecule. A molecule can become an electron carrier or electron acceptor. If the molecule only accepts electron and be done with it, for example, like this one here, NADP plus here, it accepts electron and then add hydrogen to it to become NADPH. We call NADP is an electron acceptor. Okay. However, when a compound, for example, like PQ here, which is it stands for plus 2 quinone, not only that it accepts, but it actually moves and transfer the electron to somebody else. That we call electron carrier. So plastoquinone electron carrier, plastocyanin electron carrier, but NADP electron acceptor. Okay. Look at this membrane here. Even though the image here is static because I can't find the one which is animated, these blobs of electrons carrier, they are actually moving. Okay, they move actually about a tenth of nanometer. They move in the membrane because the membrane is fluid. They move. When they get something, they move because they need to move the electron. Okay, because electron cannot suddenly jump. This is not, you are spearing something. It has to physically be closer to the target so that the electron can get transferred. Right? We'll see that in a bit. Right. So this is about the story about the bacteria as well. So whatever that you have seen earlier, this setup here, the photosystem here, that is also present in anoxygenic photosynthetic bacteria. The thing I want to highlight here is that photosynthesis is not exclusive only for plants. No, throw that knowledge away from you. No, there are many other bacteria that are actually capable of photosynthesis. Okay, so is there any difference? There are difference in the detail, but the concepts remains the same. Okay, yeah. Even look at the plants. Not all plants are actually photosynthetic. Did you know that? Some plants are parasitic. Yeah. In Malaysia, we got pokok dedalu that is parasitic. In the more temperate, I know more temperate example, we got plants like mistletoe, dodder plant. These are parasitic plants. They don't really photosynthesize. They just, sucking up all the goodness from other plants. Okay, right. So the bacteria that we're talking here that can do photosynthesis. So let me see. Does it look familiar here? Yeah. In plants, you can see there is a system like this, right? Photosystem II, photosystem I, right? But in bacteria, it can be a more simpler version of it, just one photosystem. Okay? Because some bacteria, they don't evolve oxygen and oxygenic, non-oxygen evolving. For plants, because the presence of PS2, oxygen is released. Okay, that's why we get oxygen. But for bacteria, they just want to have the energy, this thing, and also ATP. So in order to have these two compounds, one set of photosystem is enough. Okay, because bacteria is already small, whatever oxygen dissolved in water is enough. Okay, so in bacteria, the chlorophyll is now called bacterial chlorophyll. However, chlorophyll in bacteria, please bear in mind, in order to activate or in order to excite it, you need different energy. It's not like in plant. In plant, the energy needed to excite the chlorophyll about 680 to 700 nanometer, meaning that the deep red. However, for bacteria, you need P870. P stands for pigment. 870 refers to the nanometer of light energy needs to activate the pigments. Okay, so now you learn that there are other type of P. You learn so far you got P680, P700. Now in bacteria, P870. You can't see it because this is infrared. Okay, you can sense the presence of it in the form of heat. Okay, right. Yay. Not to worry, I'm not going to answer, give you have you answered today. So later, maybe in the afternoon, I'll have the entire set, all of these quizzes for you to take home and do a little homework with it. Okay, so let's put it this way. I think there's a collection of 9 because we got 9.9, right? So 9 sets of quizzes. Not only that, I want you to answer it individually, not in group. Answer it, then show me where is the answer coming from the notes or anywhere. Maybe you want to, photostate this again and then cut the small bit of it and then stick right to answer, highlight it. This is my answer. The art of finding answer, so that you know your answer is not coming from some kind of blabbing guy 7:45 in the morning. It's actually written in the book. Okay, so for example, you have this integral membrane protein of the chloroplast have a random orientation within the membrane. True or false? False. Show me. Why is it false? So maybe you want to screenshot something from the notes to show that this is not random. Yeah. Validate, verify your answer. All right? Okay. Okay, take five, because this section I'll just do in 20 minutes. Still breathing. Hour in the afternoon, 2 until 3, then you're done. Kau boleh balik tidur balik. Kan nak relax, bertenang. Right. Finish this in 20 minutes. Finish this in 20 minutes. That sounds very ambitious. The organization of the antenna system. So, on the surface of the thylakoid membrane, in addition to the integral protein, how many protein you learned just now? What are they? Photosystem II, Photochrome, and ATP synthase. Okay. In addition to these proteins, you have another structure to the membrane, which is the antenna system. Okay. Antenna to send what? To get the light. Somebody has to receive the light. ATP synthase is not going to receive the light. Yeah. But there is a special story to this antenna system. Okay. So this antenna system, in essence, it will have a number of chlorophylls per island of it, meaning that there are many islands on the surface. Let's say that this is the surface of your thylakoid membrane. On this surface, it could be like maybe 1 million islands of antenna system, and each antenna system could comprise or consist of 100 to 200 molecules of pigments. Okay, one island, 200 pigments molecule, one island, 100 molecules of pigment, and so on. Okay, and this energy will be absorbed by the antenna system for one reason, so that it can direct the energy to the reaction center of an antenna system. So even though there could be a number of antenna system on a given thylakoid membrane, each system will have one reaction center. Okay, one reaction center. Okay, and the energy that is moved from one pigment to another pigment, we call it FRET, fluorescence resonance energy transfer. Okay, and this is non-radiative. Okay, non-radiative meaning that it's purely physical, meaning that one molecule absorbs light energy and then it will vibrate, and this vibrate will be sensed by the adjacent molecule to it, and it will start to vibrate together. It's pretty much like a tuning fork. You bang a tuning fork, one tuning fork is, and then you have another tuning fork. If you touch them together, some of this frequency will be transferred to the second, and then this one will as well, right? And so on. But of course, this one will in a lot more calmer, relaxed compared to the first That is called vibrational frequency. Maybe it's mentioned in here somewhere. I don't know. Maybe it's mentioned somewhere. Ada ke vibrational frequency? At least you know I'm not scamming you. Sorry lah, I've learned this more a long time ago. I know it's written somewhere in the book, but jangan tanya aku dia line ke berapa. It's for you to read. Okay, so let's look at this. The antenna funnels, that's the keyword here. Funnels energy to the reaction center. What do you understand by the word funnel? From big to small. What about the direction of it? One way, both way, side way, multi-way, one way. Okay, so two concepts of funnels here, meaning that from big to small and unidirectional. Nobody that has reached one end go backward. No, it's not happening. Okay, so for each antenna system island, you will have, in essence, this setup of pigments molecule. Okay, I'm still not referring to either photosystem 1 or photosystem 2, but both of them kind of like this. But for simplicity, most book will say that this is the setup, general setup for photosystem 2. Okay, remember this. Photosystem 1 and photosystem 2, biochemically, photosystem 2 happened first, then only photosystem 1. The reason that it's not named that way, I don't know why scientists didn't correct this. Photosystem 1 happened to be found first. Then later on, they found out, oh, by the way, energy come from this guy first. Yeah. So photosystem 2, just written, it's number 2, but sequence, it is number 1. Right. Okay. So you got your light. Light, the energy is high. Okay. Will be accepted by molecules of pigments. This is called collectively as antenna complex, meaning that in this complex, not only one pigment or one color. There are various colors. There are chlorophyll, there are carotenoid, xanthophyll, viola-xanthine, and so on, depending on plant species. But in essence, all plant species have these things, carotenoids and chlorophyll A, B. So these will keep on passing the energy. Maybe the carotenoids, if you remember from your notes here, the first week note, where's that note? From the first week note, I think figure 9.7, you can see that for the carotenoid or beta carotene, the peak absorption happens around 470 nanometer. Okay, meaning that 470 nanometer absorbed by the carotenoids. However, this is not the energy that can excite or activate the reaction center, because the reaction center requires 680 nanometer. So 470 here will be passed to the second molecule. Now it becomes 500. And it passed to the second molecule over and over and over again until the energy becoming lower enough to hit 680 nanometer. Only this energy will activate the reaction center. So what exactly is reaction center? Reaction center is actually a special chlorophyll A molecule with a loosely bound electron, meaning that the electron can be ejected out if it gets excited. It's kind of like, remember, I give the analogy of excitation like you and your friend laughing together. Yeah, you keep tickling on your friend. She's laughing, right? She's laughing. So she's in excited state. Yeah. Your other friend is also laughing. But none of them laughing and anything coming out from their body. So they are all these antenna complex. But this one friend that's about to go and snitch to the teacher that you guys are laughing non-stop. She's the one with the denture. Get excited and the denture come out. And that come out is kind of like electron get ejected out of this. Go to where? Go to the teacher and tell the snitch on you. You have been creating a lot of noises in the class. Okay? Right. So, another thing that you need to understand in physiology about this asterisk or star. The moment you see a substance or molecule followed by a star like this, meaning that it has been excited. Excited carotenoid, chlorophyll B excited, chlorophyll A excited. The one without the star, we call it ground state carotenoid, ground state chlorophyll, ground state reaction center. Excited state reaction center, the moment it has star in it. Okay? So keep on passing and then activate or excite the P680. Okay? Yeah. So, oh yeah, it's mentioned here. For the excitation to be transferred back to the chlorophyll B, it is just not possible. Yeah, because this energy here is already low. Low energy cannot go back up to go against the energy gradient. That's why it's called funneling, meaning that P680 cannot transfer back to the carotenoids. Because carotenoid, it requires higher energy to excite it. Otherwise, it's not going to accept. Okay, it's kind of like tomorrow you tell the same joke again. Your other one of your two friends not going to laugh, and then I already know this new joke, please. Not easy to excite. You need the right switch to tickle and cause the laughter throughout the class. Right. Ooh, what's this? Oh yeah, this is the 3D model of the antenna pigment complex. So just now you saw it's written antenna complex, right? What is that actually? It's actually antenna consists of two things: pigment plus protein complexes. So in one of the island, in the island that we're talking about just now, in each island, two things are present: pigments plus protein, and then they fuse together. They fuse together to form these antenna complexes. Okay, so this is the A is the stromal side, meaning that how do I say this? Let's say that this is one antenna complex in the thylakoid membrane. This is up here is the stroma side. Down here is the luminal side. That image here is looking up the island like this. This image, the first image. So from the top, looking up here, you can see that chlorophyll, the pigments, are actually connected with all these. You see the grayish thing? These are the polypeptide. So it can be any sequence of amino acids, short amino acids. Okay. Why? Because of this. I'll show this again. Remember this dimer? Yeah. Trimer? Yeah. This thing. These are protein. These are protein. Now you see it in this form. And you can see that chlorophyll is not the only pigment present. You got what? Violet xanthine, you got neoxanthine, you got lutein, and so on. By the way, you also got lutein. Lutein is, you got it in your retina, in the macula at the back of your eyes. Okay, that's how your eyes filter the damaging UV and blue light rays. Okay, and then this is the side of it. So your pigment complex, you're looking from the side of it. And you can see that some of it actually protruding out into the stroma, some of it protruding out into the luminal side, but most of it is actually in the thylakoid membrane. Okay, they need to stay in here, otherwise all this ribbon will come out. Okay, phospholipid bilayer is very good at stabilizing things. All right, okay. Ooh, how many minutes we got? All right. Can I go another 5 minutes? Okay, 9.6. 9.6. Okay. Now you know about this thing. you know about these antenna pigments, right? So how the energy is transferred? In what manner the energy or the electron is transferred? So that movement of electron, the moment it has activated the reaction center, the P680, it will move in a special manner. That manner, we call it the Z scheme. Or if you are American, you call it Z scheme. But please don't say Z, because kau bunyi macam redneck. You know redneck? Tak tahu nak cakap kat Malaysia, kat mana nak tengok redneck. Sebab semua sini tak coklat, hitam. You know redneck? Tak. Farmers kat US tu. Mungkin sebab dia sopan, dia tak pakai sunscreen semua, lepas tu dia punya belakang tu jadi merah. To show that you are low class life, working on labor, hard work. Petani lah, macam kau. Okay. Ooh, that thing. The Z scheme, meaning that the electron, how do you say this? Remember, you have learned there's a different way to say this, different way to say this. Ground state reaction center P680. Excited state reaction center P680. Okay. So, in essence, I don't want to look up. Aku cakap orang redneck, tengkuk aku yang sakit. So, we start in here, P680. Let's see whether I can remember lesson from 12 years ago. Kalau salah, cakap salah. Tak ada lah. But who's going to validate, right? So this will be excited to become excited P680. Okay, so now this will be transferred to a series of electron carriers. Okay, so you start with phyophytin, then you start with plastoquinone A, then you go to plastoquinone B, then you go to cytochrome B6F, then you go to plastocyanine. Is that correct? Then you will meet with, what is this? What's this? Yes, ground state P700. Then you're going to go up to P700 star. And then you will go to A0. This is actually chlorophyll, but we call it A0. Then we go to A1. Then you go to iron, sulfur X. Then you go to iron, sulfur A. Then you go to iron, sulfur B. Then you go to ferridoxin. Then you go to flavoprotein, ferridoxin, NADP plus reductase. Then you go to NADP plus. Betul? Betul ke? Aku still ingat. Can you imagine I have not taught you this? Kalau aku ajar mesti kau mati. Okay. Okay, since I can remember this, I expect you to know this jugalah. Ada kau nampak aku tengok sendiri? Tak ada kan? Aku tahu je benda ni. Habislah kau. Oh, by the way, dekat sini, cytochrome B6F, it has its own structure, which we'll see later. But in essence, this is the Z scheme. Z scheme. Okay. Energy go up, then it hits the excitation, then it goes down. Cascade of reaction going downhill. Hence the word Hill's reaction, named by Robert Hill. Okay. Then it goes, tak jauh pun, 68 nanometer to 700 nanometer. So there's only 220 nanometer worth of energy difference saja. Yeah. Then it will excite the second reaction center, the ground state P700. Then it gets excited. Then it will transfer to this cascade of reaction. So Fe means iron, sulfur, protein. These are all protein, okay? All of these are protein. But bear in mind with this one thing. Not all of them is integral protein. Some of them protein on the surface. For example, this thing, plastocyanine here. Plastocyanine is this guy. It's not in the thylakoid membrane. It's dekat luar. And this ferridoxin, FNR, and also NADP plus also in the stroma. They are not necessary in the integral membrane. Integral membrane protein only 4 for sure. Okay, the one that you learned just now. Okay, and then get excited 700, pass down to chlorophyll A, and oh, by the way, A1 got a name actually. The name here is, let me see, can I remember that? Wikipedia, please. Phylloquinone. Thylakoinon. Transfer to iron sulfur X, iron sulfur A, iron sulfur B. Okay, right. Sometimes you will see in the form of plastoquinone A, plastoquinone B. That is simply the reduction state of it. If you learn chemistry, I'll give an example for plastoquinone. Plastoquinone, it's got three states of it. So plastoquinone is oxidized state. Oxidized state. Then you have plastosemiquinone. So the symbol becomes like this because it only have one electron coming to it, but the moment it is filled with hydrogen, it becomes plastoquinol. Or I think in the book it's called this PQH2, plastohydroquinone. So this become pH2. This is only PQ. No charges, receive one electron, receive two hydrogen. Still similar molecule, but now the molecule with different state of charges or reduction level attached to it. Okay, all right. Since I already said plastohydroquinone, maksudnya kau kena ingat lah kan? I think it's mentioned here somewhere. Okay, all right. Okay. Oh, lupa kat sini. By the way, there's something happen in here as well. So this is why you got your H2O, right? You got your H2O got split. You got I think 2 electron for hydrogen. Is it for hydrogen? Oh, betul? Oh, oxygen. Yeah. This is where you got your oxygen. 2 molecules of water is split because the moment it gets excited, this thing contains hole. Okay, this hole needs to be filled in by somebody, and who is that somebody? This thing steal electron from water, and then the water is oxidized. Because water is oxidized, H2O is split. When it is split, that's why you got oxygen. That's plus 4 hydrogen. Okay. Yeah. In case you don't, and finally, I just want to clarify on that H plus. H means, if you only H, it means hydrogen, 1 electron plus 1 proton. If it says hydride, means hydrogen minus, no proton. If it says proton, it means hydrogen plus. So 3 levels of hydrogen we are talking about here. Are you talking about atomic hydrogen, like in the case here, or hydride of hydrogen, H minus, or proton of hydrogen, H plus? The splitting of water producing H plus, proton of hydrogen. The reduction of plastoquinone is with the addition of atomic hydrogen, right? The hydride of hydrogen, H minus, this will you learn later. Some, not all molecules accept that, okay? That is all. Still breathing? Still breathing? Okay, so, okay, that's all for now. So I'll see you again in the afternoon, okay, at 2. So we'll have another 1 hour. I think we can finish until the rest of it. Then I'll give you quizzes and also the summary of this thing, okay? It's a good thing orang tak ramai, semua orang dapat nota warna, right? Okay, any question? Why life so hard? Don't ask that. Kat mana ya? Nanti we'll make announcement in the Google Space. Okay? Kalau tak ada, we'll just go to bilik seminar. Payah sangat kan? Alright, okay.